So I'm still a professor at McGill University. I'm trained as an infectious disease microbiologist. So I have 30 plus years experience as a faculty at McGill, but I began working with this tiny garage-based company in 2003, just as they were starting to use the Nicotiana benthamina transient transfection platform. And I then worked with them as I was on their scientific advisory board, and then became a collaborator. So we held grants together, university industry grants.
Yeah, very good.
I got to know the company scientists very well. And then in 2009, we did our first human study with a pandemic vaccine, influenza. And this hand injected the first dose of a plant-made vaccine into a human being. So
it was you yourself,
me myself. But I mean, it's nerve-wracking, right? Any phase 1 human trial is nerve-wracking. If the first patient had sneezed, I would have taken him home with me to watch over him. But then I became more and more involved in the company. And in 2017, I got permission from the university to stop seeing patients. Basically, I traded my clinical role at the university for the medical officer role at the company. So I am an employee of the company, but I am a 50% faculty member at McGill University, where I do all of my teaching. I have my office and my lab, and we still do a good deal of the basic science translational research in my lab.
Do you also refer to the technique as molecular farming, or is it just in Germany that we call this? So how do you call this?
Yeah, I think one of the important distinctions, particularly for Europeans, is that the plants we are using are not genetically modified organisms. These are not GMOs. Yeah. The plants are transiently transfected by a bacterium that is very adept. That's what it does. It infects plants. So we've taken the bad parts out of the bacterium so that it no longer makes the plant sick, but we have allowed the bacterium to deliver the DNA that, in this case, codes the spike protein into the plant cells very efficiently. And that DNA remains outside of the plant genome. It does not integrate. The genetic information is DNA.
Thank you.
Which is then transcribed into RNA, mRNA. So the mRNA vaccines start, here's the DNA, here's the mRNA, here's the protein. And so what we do is we deliver the DNA for the spike protein, which makes the RNA, which then makes the protein in the plant.
In the plant.
So the proteins in the normal virus, the spike protein is in a trimer, 3 molecules together, and that happens in the plant cells. Those trimers move to the surface of the plant cell, where they assemble in very dense areas, and that concentration of the spike proteins drives the membrane to make a little vesicle, which pops off the surface of the cell as a virus-like particle. So all of that happens spontaneously. So what we get are these little 100 to 150 nanometer virus-like particles that have a plant envelope, but on the surface, they have a very dense collection of the spike protein trimers, just like the virus. If you take a picture of our virus-like particles and a picture of the spike of the SARS-CoV-2 virus, you can't tell the difference.
Cool. So one wouldn't know that the product in the end has been developed within a plant. One would not be able to distinguish the origin.
Well,
Correct?
well, if you were very clever, you could look at the envelope lipids, which are unique to a plant.
Okay.
Many of them.
So, okay, there are
unique to a
ways
plant.
to see if this is originally produced in a plant or not. But let
Correct.
me go back to the question. So, how do you actually refer to the technique? Is molecular farming wrong then? Or
Well,
do you have a
no, I
different
know
wording for it?
many people use molecular farming.
Molecular farming.
And yeah, I mean, we call it, it's a recombinant platform, right? Because the proteins are recombinant. The spike protein is recombinant, and it's based on transient transfection. And so, yes, that is what many people call molecular farming.
Okay. So, well, then I will go further more with molecular farming because I think this wording is more
People
easy
understand
to understand. Yes. And my question now to you is, now looking back to the history of molecular farming, how important do you think is this achievement which you have now in the whole history? Well, this is a milestone, definitely, but what does it mean for the progression of molecular farming? Can you say something to this?
Well, plants are plants are eukaryotes, right? They are much closer to us than, for example, a yeast or a bacterium.
Okay.
And so many of the other recombinant platforms, for example, the hepatitis B vaccine is made in yeast. What that means is the plant's way of making proteins is closer to ours than the yeast or than a bacterium. And so the proteins are not identical to, but they are more similar to human proteins. The plant also, if you think of making a biopharmaceutical, right? And you think of dividing it into two stages. The first stage is the making of the protein, whether it's a spike protein or insulin or an antibody. And the second stage is purification, right? So upstream, downstream. Downstream for us is really very similar to all other recombinant protein platforms, right? You use centrifugation, filtration, and other standard pharma technologies for getting rid of what you don't want. Okay. Upstream is completely different.
Yes.
Because the upstream in other biopharmaceutical manufacturers are basically big fermenters, right? It looks basically like a brewery, right? You have these big stainless steel fermenters that are carefully controlled, in which you grow the cells, yeast or bacteria or other cells, insect cells in some cases, and then those are called bioreactors, right? They're making a biological pharmaceutical. Our bioreactor is the living plant in a greenhouse. And so it is the upstream is completely different. And so what that means for us, and I think what this means for other people who might want to do the same kind of thing, is that all you need is a carefully controlled greenhouse. And so you can imagine the central facility where we do all of those downstream things is essentially the same. But you have greenhouses that are sitting around the central facility that are making influenza vaccines, SARS-CoV-2 vaccines, antibodies, monoclonal antibodies for cancer therapy. In 2014-15, we had a government contract to make the Ebola antibodies for the ZMapp cocktail that was used in West Africa. right? And because plants make proteins that are more similar to human proteins, the ability to basically expand your greenhouse to whatever size you want, and then funnel that material, which we call biomass, into the central processing facility is potentially very cost effective.
Now that's the point. It's good you mentioned that. Also the ZMAP thing, because ZMAP has been approved, but that was no vaccine. That was just for
antibodies.
Antibodies, right? So since this is the first time a vaccine has been approved, I do believe that this is also a big milestone in terms of how in future molecularly farmed or plants which have been produced through molecular farming can be accepted worldwide. So I think this is a milestone which is important, because until now no one would have thought that this would ever be possible, that a vaccine would, yeah, well, that's what I think. What do you think? I know I do understand all the advantages of molecular farming. I would like to know from you personally, what does this milestone mean now for you, that this has been approved? Or did you always know that this would be approved? Is this no surprise for you?
Of course not. Of course, well, no, not a surprise, but I mean, we were hoping it would be approved. And well, I won't give you any names, but about 7 years ago, we were invited to a very powerful scientific interview, and basically some very powerful people in the United States were considering providing us with government support for the facility in North Carolina. And one of the most important people in that group, we gave them a tour of the facility. And one of the most important people in that group leaned over to me at one point and said, when your vaccines fail, you can always grow flowers in your greenhouses, which was A, insulting, and B, it was skeptical, highly skeptical.
It's stupid.
Well, no, not at all. It was a new technology, and we were the first ones doing it at a large scale. And the facility, and by the way, the facility that we were touring was built with US government dollars. It was built with DARPA financing. Mm-hmm. And this person was heavily involved in the DARPA financing and was not in favor of our technology. We presented again to that same group of scientists 2 weeks ago, and that same individual remembered what he had said to me, and he basically said, "You've done it. You have convinced me that this can work." And I have to admit, that was one of the nicest things that happened, because the chair of the committee, who had always been supportive, he basically said, if you can convince this person, we're really impressed, because he's not easy to convince. So we've gone from, I think, a high level of skepticism in the feasibility of the approach to demonstrating that it can work. And we've had to invent everything along the way, right? We have a robot in the North Carolina facility that picks up several hundred 12 to maybe 18 centimeter high plants, and it turns them upside down, puts them into a container with the bacterial solution, clamps a lid on the top, so it's like a pressure cooker.
Okay.
And then it sucks all the air out of the tank, so that there is a vacuum applied to the roots of the plants.
Okay.
That allows the plant leaves to collapse like a sponge, right? Remember your high school biology.
Totally.
The roots are connected to the leaves through the plant vascular system, And then when you release the vacuum, the leaves re-expand like a sponge, and the liquid with literally probably hundreds of millions, if not billions of bacteria are sucked into the entire vascular system of the plant. So every cell in the plant is transfected all at once. And so that takes about 2 minutes.
2 minutes.
And then the top comes off of the pressure cooker. The robot goes back in and lifts the plants, hundreds of plants out at the same time, turns them right side up again, and then they go into our controlled greenhouse to grow for another 4 to 7 days.
4 to 7 days.
That's all.
Okay, wait, let me go back. So you have 2 minutes for 100 plants, and then another 4 to 7 days. When do you start? Well, you cannot obviously do it when the plant has just come up. You have to wait for a couple of weeks until the plant is big enough.
About 6 to 8 weeks.
6 to 8 weeks.
Yeah,
After
when the plants are about that high.
Okay.
About 18 to 25 centimeters high.
So 6 to 8 weeks, you have a plant. You have hundreds of them. You put them into 2 minutes of this excellent
bacterial solution.
Bacterial solution. And after 7 days, you have grown in the leaves the proteins which you need. And that's it.
The virus-like particles are in the leaves.
Mm-hmm.
And then all we do is we chop up the leaves and digest away the plant wall, the cell wall. Yeah. So cellulose and pectin make the plant rigid. We just digest those away, and the virus-like particles float free into the liquid.
Excellent. So if this is so easy, why did it take so long?
Number one, it took our little company with 750 employees 2 years. Okay. And it took several major companies with more than 70,000 employees 12 months. So you can do the math. Yes. And the other reason was that most of the other vaccines that came very quickly onto the market were already funded by CEPI to produce a MERS vaccine.
Okay.
And so when SARS-CoV-2 arrived, CEPI asked those groups and those companies, if we give you more money, will you start making a SARS-CoV-2 vaccine instead of a MERS vaccine? And of course, if somebody offers you more money, what do you say?
Yeah, sure.
You say yes. So most of the vaccines, so the Oxford group, the group in Australia, the BioNTech, CureVac, most of those other vaccines that came very quickly had already been working with CEPI support on a MERS vaccine.
Now, and this actually means that now that you have been approved with this technique, in future you would also be considered for support if it hopefully not comes up again and again, but at least we have some new variants. We have Omicron and others to come. And I assume that your approval has been for a mixture, which is probably for Alpha or Delta or whatsoever, but not yet for Omicron. Couldn't be because Omicron appeared just now. So how quick can you be actually to produce a new injection, a new dissolution with bacteria to make a new vaccine? Yeah. What is the time scale on this?
Well, so there are many ways to answer that, and there are dishonest ways and there are honest ways.
I go for the honest way.
Okay. Well, but you have to understand the difference. So we can make a candidate vaccine at the bench in about 2 to 3 weeks, maybe 4 weeks. The yield might not be great. The protein stability might not be great. There are many considerations. With influenza, our virus-like particle vaccines for influenza, we have almost 18 years of experience making hundreds of different influenza constructs.
Well,
right?
wait a minute, but not with molecular farming, or there isn't.
Yes,
No.
yes.
Oh, yes. Oh,
Yes,
I wasn't aware.
we were this close to having an influenza vaccine licensed in 2019 when SARS-CoV-2 came. So there's a major paper published in The Lancet in early 2020 that describes the phase 3 studies that we did with our quadrivalent influenza vaccines.
I will, 2020?
2020.
Yeah, that's good.
I'm the first author, so it's
then I will find it. Yeah.
Ward BJ, and it's in The Lancet. So
Thank you.
we will go back to influenza quite quickly.
Once
SARS is over.
And now is the question, and do you think, is it going to be more easy now to get the next vaccine and influenza approved, now that in general you got the skeptical persons on your side to believe that this can actually work?
We hope so.
Yeah.
Right. The first one is always the hardest.
Yeah.
And so we've now demonstrated that it can work and that it can be effective in human beings, a plant-made vaccine just in general. And so, yes, we hope that this will make other authorizations for other vaccines easier.
So this is like a symbolic thing, actually, because maybe this vaccine will not help for the future, for the next variants to come, but symbolically.
Well, no. So you asked that question.
Yeah.
The honest answer is it takes about 3 months to start from a sequence. So we, just as a proof of concept exercise, we started working with the beta variant when it emerged in South Africa. And so we actually have a beta variant virus-like particle candidate that we will be testing in a limited number of people as a booster, starting in about 2 weeks. And the reason that we chose beta was that at the time, it was the most genetically different variant that had emerged. Of course, now Omicron is even more different. But I mean, the problem with these variants and the speed with which SARS-CoV-2 is changing is that if it takes you 3 months to make a vaccine at high yield, and then it takes you a month and a half, 2 months to test it in human beings, so now you're at 5 months. That's the lifespan of one of these variants, right? We're already seeing Omicron start to disappear.
Right.
So you could argue, and we did. We had huge pressure from some people within the company and outside of the company to make a Delta variant vaccine, and we resisted. We said that doesn't make sense, because Delta is much more similar to the ancestral form that came out of Wuhan than the beta is. Let's take the most genetically different one and try that. Well, Delta is in the process of disappearing. It caused a huge problem in India and North America, Latin America. Same with Gamma. Gamma took off in Latin America. It caused a huge problem, just like Beta did in Africa. But they're both gone or going. And now we have Omicron. So what's going to happen next? We don't know. But what we do have is our molecular biologists have made many different constructs of Omicron. So they target Omicron. So we're testing them in the plants, and we will test them. We have started to test them in animals, but we will not make the commitment, which is very expensive for a small company, to do something that's called make a master seed bank to then test in human beings. Those are extremely expensive processes. So we are not committing to doing those very expensive things, but we're doing much of the work we need to in the event that Omicron or one of the sons or daughters of Omicron becomes the endemic virus,
Okay.
in which case we will be ready easily within several months to produce a candidate Omicron vaccine that we can quickly test in human beings, because we will have done much of the work and then we'll just have to verify, right?
And probably you
So
can also then combine it together with the influenza in case you get that proved, right?
correct.
So, yeah.
That is on, we've already done animal studies showing that our vaccines are virus-like particles when you give them either here and here or in the same arm in an animal, in ferrets, that you get a good response to both.
In what? Excuse me?
Ferrets?
What is that? Sorry.
They look like a weasel.
A weasel.
They look like a weasel. It's actually a European animal that I'm not sure it even exists anymore in the wild.
Oh,
That's a ferret.
I think I know what it is. Wie heißt das denn? Yeah.
People keep them as pets.
I think it's a Frettchen. I will look it up. Thank you.
Some people keep them as pets.
Yes.
They're very loving, they have very sharp teeth, but they can live for 10 to 12 years. So many people keep them as pets.
But you don't have a ferret as a pet.
As a pet? No, I have dogs.
Okay. So going back to what has been the biggest challenge to you now personally or within this development of the vaccine?
Time pressure and the number of people that we have in a relatively small company. Each of us has to wear many hats to get things done. So for me as an academic, it has been fantastic because I've been able to see the whole process from beginning to end, and participate in all of the different stages. I was not born in industry. Right? Academics always think that people in industry, are not as smart, don't work as hard. I mean, there's a divide between universities and industries, and the last 20 years of working with my colleagues at Medicago has proved most of those impressions just to be wrong. The people in industry are just as smart. They work just as hard. They exist with timetables that mean something instead of academics who live, with tenure, and so we don't really care at some level. And so it has been a high pressure, exciting, but tiring. So, when I wrote to my family and friends last Thursday, I said, we are really tired, but we're going to go to sleep smiling tonight. Right? Because we're tired, but we're excited. It has been a wonderful experience in many ways, but challenging, because everything we're doing is new. It's not a standard pharma technology.
So
many surprises.
Okay, now talking about since this is no standard procedure, what is the perception of people outside? Now, not only the people involved, of course, to the university, everyone there will understand this, but people outside, like, I don't know, friends, people who you talk to who have no background in biology, academics, do you encounter that they have prejudices not against the industry, but rather against the whole thing that you're growing medicaments and plants, and then in tobacco? Is there something you
Oh,
encounter?
that's something that I have to correct.
Oh, please.
It's not tobacco. It
Bentiana,
is
nicotiana is not tobacco. Excuse me. Please correct me.
Nicotiana is the family. It's Nicotiana benthamiana. Tobacco is Nicotiana tabacum.
Ah,
And Nicotiana benthamiana is an Australian cousin of tobacco. So it's in the same family of plants, but just in the same way that a chihuahua and a wolf are also in the same Canis family, but they are clearly very different animals.
thank you.
And so Nicotiana benthamiana is a weed that grows in Australia, and nobody smokes it or chews it. It's just a weed. But it grows very quickly. It has an immune system that is weaker than many other plants, and that allows it to be transfected and stay healthy much better than almost all other plants. That's why it's used in many molecular farming exercises. It is probably one of the commonest plants used by plant molecular biologists for that reason exactly.
Thank you very much for putting this clear. And I have to ask exactly to this before we go on, because I think in Germany we really call it tobacco plant, but it might be wrong. Totally.
It is absolutely wrong.
It is absolutely wrong. Thank you.
The Nicotiana benthamiana grows to be about this tall.
Yeah.
Tobacco, like Nicotiana benthamiana that's used in the tobacco industry, grows to be almost as tall as I am.
Yeah, or even taller.
The leaves for Nicotiana benthamiana are about this big, while the tobacco leaves are massive. They're huge.
Okay.
So yes, they are in the same family, but
the same thing. Thank you very much. That's very important.
If you're going to be in a small room, with a chihuahua or a wolf, which would you choose? So we clearly understand those differences, but many people just assume that it's tobacco, but it's not.
Which is maybe also because it's the idea is really nice that instead of making you sick by producing cigarettes, it might actually heal you by producing a vaccine. Which really
yes, there are some ironies there.
There are.
But really, honestly, it's a plant of convenience because of its biological characteristics. In just the same way that most or many proteins are expressed in a specific kind of yeast for recombinant protein production. That's just because that's what people have always used. Yeah. Right? There are thousands of different yeasts, and nobody has tested all of them. And so it's just easiest to use the same yeast that everybody's used before.
Did you experience concerns and prejudices? If you tell someone, well, I have a medicament which has been produced in a plant, you will probably have very different impressions associations that maybe you're making a nice yoga plant, which you understand where this is going. I would like to know what kind of prejudices do you encounter and what do you respond to that?
Well, obviously, it can go both ways. There are people who misunderstand that it's a tobacco product, and there are people who think that it's a vegan product.
A vegan?
And
excuse me, I don't know the word.
Vegetarian.
Vegan, yeah.
Right. So, and both of those things are incorrect.
Right.
And so, yes, we do spend some time talking to people and making sure they understand that the vaccine, the virus-like particle vaccines that we make are plant-derived. They are produced in plants. And yes, some people are biased in favor of that, And if they think it's a tobacco plant, then they are biased against that. So we have to deal with both misconceptions.
That's what I was thinking. I just wanted to know whether my clue was right. Thank you. Did you encounter any surprises, good or bad, in the past? Is there anything where...
I think that we were surprised. Well, so you know that our COVID vaccine is delivered with GSK's adjuvant.
GSK. I have to look that up again. GSK what? Wait, excuse me.
The adjuvant is ASO3.
ASO3. Okay.
Right. So our vaccine is delivered with that adjuvant, and one, I think, surprise was that the protection against SARS-CoV-2, like many viruses, in the short term is driven by antibodies, right? Antibodies are a good thing.
Yeah.
Right? Having lots of antibodies is a really good thing, and having lots of antibodies makes it more likely that you will have cross reactions to variants, right? So we know all these things. But the real immunity against viruses, most viruses, is cellular immunity. That's the long-term memory. So there are many people who are completely protected against many viral diseases, not because they have good antibody memory, but because they have good cellular memory.
And this is different from person to person?
Almost everybody. So let me put it this way. There are some people in the world who cannot make any antibodies at all. Zero. Those people do not die from viral diseases.
Okay.
There are very few viruses where antibodies are necessary. However, there are some people in the world who have no T cells, so that important part of the cellular immunity, and those people die all the time from viruses.
So it is actually about the ability to produce T cells, whether
Correct.
you... Okay. Now go back to what your surprise were. Thank you. Yeah.
So our virus-like particle vaccine, without the adjuvant, makes really good T cells. And with the adjuvant, it makes much better antibody responses. And so the two of them together, we get both antibodies and T cells. So we think that that's going to be very important for how long the immunity lasts, and also protection against variants.
Now, then let me ask me exactly, in comparison to the other vaccines which are out already, did you ever make some kind of study to compare what how good is the T cell production and antibody production with Medicago's vaccine in comparison to BioNTech, to Johnson & Johnson, and all these others? Does something like that exist, or is that something which is coming now?
So we have not done any studies like that because the vaccines are controlled by governments. And of course, if one company goes to another company and says, "Can we have some of your vaccine, please?" the answer is no. However, several countries have done head-to-head comparisons, right? So they've compared the Pfizer-BioNTech to the Moderna, to the AstraZeneca, to the J&J. And actually Novavax, their vaccine, has been included in some of those studies. And what's very clear is that antibodies, right after the vaccination, the adenovirus vaccines are about here. The mRNA vaccines like Pfizer and Moderna are about here. And the protein adjuvant vaccines like Novavax's and ours are here. So about 1.5 times what you see from somebody recovering from disease, about 3 to 4 times what you see, and our vaccine is 10 to 14 times higher.
But that's amazing. Why is this? Why, is it because of the plant? Has it something to do with, no?
It's because of the virus-like particle and the adjuvant. So protein adjuvant vaccines are very good at making antibodies. Very good. And in our case, those antibodies last for a long time. We know that. We've actually published that. Those antibodies also are very cross-reactive. We've also just published that. What is different is the cellular immune responses. So, for example, some of the Chinese vaccines, for example, that use the inactivated virus with an adjuvant, they make very little T cell response, very little cellular immunity. The adenovirus vaccines and the mRNA vaccines, they make good T cell responses. They do. What's really interesting is that so does our vaccine. Our vaccine makes very good cellular immune responses, even though it's not producing the protein like the mRNA or the adenovirus vaccines inside a human cell, which is normally how you get T cell immunity. The virus-like particle, we think, tricks the human body into thinking that it's a real virus because it looks like a virus. It's the same size, it's the same shape, it has the same surface molecules, and we think that that fools the immune system into making a strong cellular immune response. And so our vaccine, so that was a surprise. It was a happy surprise.
Yeah.
But cellular responses are intrinsically cross-reactive. Right? Antibodies can be very, very specific. Right? I'm going to kill Omicron, or I'm going to kill Delta, or I'm going to kill Gamma. Right? Some antibodies can bind to all of them, but antibodies tend to be very specific. Cellular immunity is not specific. It tends to be against conserved epitopes. So it tends to be directed against little pieces of a protein that are found in many different viruses. So yes, that was a surprise, and it was a lovely surprise.
That is a great surprise. And it also actually means that it has a great chance of helping against other variants to come. Because
correct.
Yes, that is so. So I have to go a couple of questions back. How many plants do you need to produce one doses of a vaccine?
That is, I've never, I don't think I've ever done, or anybody has ever done that calculation.
No worries.
Because we talk in terms of biomass,
Yeah.
and also the amount of protein that is produced by the plants is, we're steadily improving that.
Okay.
So
Okay, but biomass means kilogram, and kilogram is something like I can understand. So how many, or what do you mean by biomass? Excuse me.
Yeah, it's biomass. It's so the ground up plants.
Okay, so how many kilograms?
I do not have. I can send that to you by email because I don't have that in my head.
No worries. It's just for I'm just curious. Is it like as a room full of plants, or is it a I don't know.
Oh no. It's no. I do not know how many doses of our vaccine come from one plant. But
from one plant you can make more than one doses.
Oh yes.
Yes. Okay. No. This is enough for me to understand. So okay, it's not you need
many
dozens of plants for one dose.
No. Many doses will come from one plant.
Okay, well then you don't have to send it to me. I needed this to know whether you need a football campus full of plants to produce 10 vaccines, but it's exactly the other way
No,
around. You get
it's the other
lots
way
of.
around.
Okay, cool.
Yeah.
This is
So
actually a good.
when we make a lot in our at one time, so when we infect a group of plants, we will typically get between 1 and 1.5 million doses from a lot.
Wow.
So
But
so
but this must be so much more cost effective. Your vaccine should be so much cheaper then, in case the Cotiana benthamiana is not a really expensive plant. I believe it must be so much more cheaper to produce it with molecular farming than in the normal way in a bioreactor, or
so we all believe that's true,
yeah,
but the first time you do anything,
of course.
you have to invent everything. And so I think that in a decade from now, when this platform when this becomes standard, I think that the cost of goods will be low compared to other, but at the beginning, that just isn't true. And so one has to be very careful about saying that, this is a fantastically inexpensive way of making vaccines. I think it will be, and I think that it can theoretically be modular, right? The Germans and the Dutch and the Japanese are very good at making fully automated, robotized greenhouses, right? Well, this is one of our visions, is that it does not cost as much to build one of our facilities, right? Because the upstream is a greenhouse.
Right.
And so the raw materials are dirt, water, and sunlight.
Right.
Right?
Thank you so much, dear Brian.
Okay.
That was great. And thank you for explaining it in a way that I could understand. Thank you.
Okay. Thank you for your interest.
Thank you. Bye bye.
Bye-bye.